we live through these uncertain times, we hope as a community to collectively support one another and share our knowledge and voices. Welcome to Metabolic Studio's second Interdependence Salon. We continue in this format what we began in 2016 at Metabolic Studio in downtown Los Angeles, to come together to build trust and share time as we reimagine the world. You never change things by fighting the existing reality, said Buckminster Fuller. To change something, build a new model that makes the existing model obsolete. Achieving this interdependence takes both seeing and doing, both vision and action. A new collective vision is needed, centered and prioritized around the common good and community. Today, we're welcoming James Oliver, a radical mycologist whose practice advocates for fungi, some of the most neglected and poorly understood organisms, being not plant nor animal, but its own kingdom. We're thankful to have him working with us as part of the Farm Lab team at Metabolic Studio. With us, he works to combat climate change and mass extinction through the study and applied science of living systems. Together, we are focused on heavy metals, especially lead, that's a part of the industrial corridor where our practice is situated. Today, we're going to be in conversation with Diego Zapata, Metabolic Studio's resident biologist with a special interest in investigation and investigating the resiliency of urban ecological systems and their conservation and habitat fragments through the communities of Northeast and East LA and on the LA River. These two young men have been working with us on Undevelopment 1, converting a Vertelli's tow yard into a place that is sanctuary for living systems. So I'll go ahead and let them pick up the conversation from here. They'll speak for a while and then we'll segue into questions.
Thanks, Lauren, for that fabulous introduction. As a part of the Pharma division at Metabolic Studio, I have worked with James on everything from the daunting tasks of remediating decades worth of lead and petrochemicals from the soil along the LA River, the construction of hugels under the guidance of our hugel queen, Mcadams, to the identification of inconspicuous and unknown mushrooms known as LBMs. That's little brown mushrooms for those of you who aren't following along. He's a brilliant mind, and I can honestly say that despite my protests and rigid allegiance to plant life, James has really transformed how we look at mushrooms. I, for one, have grown to really respect all those humble food caps and turkey tongue mushrooms, and I hope everyone tuning in can leave this along with a similar love and fascination with the all-powerful mushroom. With that being said, James, if it's okay with you, I would like you to take a quick moment to get everyone on the same page regarding our dynamic as peers and friends. Yeah, I know that like we often fight about which kingdom is superior, plants and mushrooms, but like, I don't know. Do you have anything to say about that?
Yeah, I mean, both Diego and I kind of have an ongoing banter in Farm Lab where we, debate like, what organisms came first, fungi or plants. And I have crazy theories about how plants are just like, inverted mushrooms or inverted fungi and things like that. And we, like Diego said, we sort of go back and forth, but ultimately I think, all jokes aside, what we do look at our relationship as is something like mycorrhizal fungi, is this symbiosis and solidarity between two organisms that work together, plants and fungi, to restore our planet and support all living systems.
Yeah, so in regards to that, what are mycorrhizal fungi?
Yeah, so mycorrhizal fungi, the term myco meaning fungus, and rhiza meaning root, represents an organism that bonds with the plant roots. So mycorrhizal fungi in some cases do produce mushrooms, but oftentimes they just form mycelial networks underneath the ground. So oftentimes we don't see them. But there is this exchange between them where the plants will trade their photosynthesized sugars with this fungal network underground, and the fungal network extending their plant root system by 10 to 10,000 times will gather resources such as water, phosphorus, nitrogen, and sort of trade for these photosynthesized sugars. And in addition, the relationship also strengthens their ability to resist erosion, resist plant disease, resist salinity, and also resist the toxic effects of heavy metals. And also something we'll be talking about today is the cleansing of soil from heavy metals.
Right, yeah. I mean, that explains it all. Our relationship borders on symbiosis and parasitism, and like it's always a like give and take, and so I apologize if there's a little fighting going on during this interview. But we're all friends, and he's one of my most esteemed peers, and I love him very much. So let's just continue on with our interview. So under the theme of interdependence, I think it's important to draw some junctures between the work you do and the studio's goal for regenerating living systems. What's the symbolism of mycorrhizal plant symbiosis and interdependence?
Yeah, that's a great question, and in my opinion, a great example of interdependence. Fungi in general present this theme of interdependence throughout evolutionary history, and that they perhaps co-evolved is not the right word, but definitely held hands with other organisms through previous extinction events with insects, with plants, and with other organisms on Earth. But particularly, if we're looking at mycorrhizal fungi, mycorrhizal fungi are obligate symbiotes, which means that they do not exist without plant roots. They don't form their own mycelial networks independent of plant roots. So you have that one symbolism of interdependence, but also alluding to our evolutionary history on this planet, mycorrhizal fungi and plants share a sacred and ancient bond together in that during an extinction event where an asteroid impacted the Earth and blew all sorts of debris into the atmosphere, scientists have wondered, how did plants survive this event, considering that the skies were darkened with the amount of debris and being unable to synthesize. And through this event, all plants that didn't form relationships with mycorrhizal fungi went extinct. And it was only through this interdependent relationship that the fungi, which needed the plants to begin with, provided these plants with nutrients during this time in which they couldn't photosynthesize. And this is a 500 million year old relationship, and it actually hasn't changed that much. So that's why today 90% of all plant species form mycorrhizal relationships together.
Yeah, no, plants definitely depend on mycorrhizae and vice versa. The work FarmLab does is primarily centered along the Ella River and its restoration into the vein of life that it was and is for a region. As such, I think it's important to understand the roles fungi have here in this unique ecosystem. What are the different ecological niches that fungi fill around the Ella River?
Right, so the general breakdown of sort of fungal niches in ecology are you have your what we call saprophytic fungi or decomposers. These are the fungi that take dead material and transition them back to life. So they're kind of these transcendent organisms that bridge the ends of life to the beginnings of it by taking dead organisms and returning that to soil and available food. So you have your decomposers that are sort of churning debris and making that available to organisms in and around the river. Another class of ecological niche is endophytic fungi. Endophytes are actually fungi that live within plants, so they're a little different than mycorrhizae. They don't live within the roots. They kind of live more within like the plant tissue itself of like the stems and leaves, but they have this mutual relationship where the endophytic fungi oftentimes produce a slew of medicinal compounds, in some cases that attribute some medicinal qualities of our traditional plants, but we certainly know that they provide protection against pathogens, and all sorts of other things. So you have this role playing along the river, sort of balancing and acting like an immune system and keeping pathogens and even overgrowth in check. And then the next ecological niche is Mycorrhizae, which we kind of talked about, which is these sort of grand symbiotes of root systems that cycle nutrients and water and prevent erosion and build soil. Ultimately, they create this compound called glomalin, which is a unique compound for people who are into vermicompost. It's abundant in vermicompost for all you worm lovers, but glomalin is basically what makes sort of dust turn from soil, or sort of what makes dust turn into soil. It kind of clumps things together and gives soil this quality we call aggregate stability that makes it really resistant. And then lastly, the other niche we have are parasites. And this kind of gets a bad rep. Parasites, you kind of get like a feeling. But parasites actually do play a really important role, especially when it comes to, we oftentimes are averse to them because they destroy a lot of our crops, but oftentimes that's because we have a monoculture. And that sort of speaks to the role of parasites, is that when there is an abundance of, or an imbalance of too much light, for example, in insect populations, these parasites are key in sort of maintaining a sort of biological diversity and balance in the ecosystem. So that's a big summary on the different ecological niches they might play in and around the river.
Great. Implicit in interdependence is the fact that all life on Earth is interconnected. What we do here in LA now has implications for ecosystems on the other side of the world later. How does the remediation of the LA River intersect with the well-being of other parts of the world, like the Pacific Ocean?
Certainly. So the Los Angeles River, for those of us who are from LA and have been paying attention to the river, does feed out into the port of Long Beach, I believe, meaning that all of the runoff from the street, all of the things we dump into the river, all of the trash, all of those things, plastics, chemicals, heavy metals, even nitrogen, which can, is generally good for organisms, but in abundance causes toxic algal blooms. So all of these flow out into the river. Out into the Pacific Ocean, which is the largest ocean in the world. So speaking about fungi and the Pacific Ocean and how the LA River all connect together, is that definitely the work within the river beyond just bending the river, but sort of river revitalization in removing the concrete jacket like we have in the soft bottom and things like that. These systems of organisms are actively cleaning the water and preventing these things from going out into the ocean. And so the work we do here has a downstream effect into the ocean and also into the Pacific Ocean. Just to kind of touch on fungi's role there, there's also the prospect of what are fungi doing in the ocean. It's only recently, within the last few years, that there's been talk about how fungi basically perform the same role they do here on land, and that the ocean floor is full of mycelial networks that are decomposing and breaking down organisms. But also, we can look into, how are the pollutants affecting this system? And we've all heard about how mushrooms can help save the world and break down oil and things like that. How can we support the organisms in the ocean so that they can be better equipped, not only from us stopping the pollution, but also empowering them to deal with it as well, and studying them to apply that knowledge.
Now that we've talked a little bit about the importance of these ecosystems and the remediation, I think we need to discuss how exactly FarmLab is planning on employing biological mechanisms to sustain and propagate life in very impacted parts of our world, like the LA River. How exactly do mycorrhizae and plants work together to remediate soil of lead and other heavy metals?
Sure, that's a great question, but I think it's just a little important before to just maybe talk about how what we're dealing with and where it comes from, just to touch on it, and sort of what the practices, the standard practices are. So in the LA River at the Metabolic Studio, we're looking at the legacy of the petroleum industry and lead-based products, and also leaded fuel. So within sort of the first 5 feet of our soil, we're looking at various degrees, some very high, almost definable as a super fungicide, and some low but still toxic of lead contamination. And then if we go further down to 15 feet, down even to the water table, we're looking at these oil plumes. And particularly, we've sort of sourced that to, in the state park, the train yard used to be there, as well as Standard Oil and some neglected oil tanks. And so these are kind of where these things are coming from and how we're thinking about dealing with them. And talking about dealing with them, most of the conventional practices for dealing with lead and oil are actually to kind of introduce solvents and detergents and strong acids into the soil, which is very cost effective, very cost intense, not cost effective, as well as a very intense carbon footprint. And ultimately, the result is that the soil that you're taking, in the end, what you're looking at is really no longer soil. It kind of has to be done. It's sort of been transformed into inert material, in that the years of process of seed and pollen, of glomalin, of decomposing, and things like that that have built up in these layers are all being washed away with these intense chemicals. So though we're cleaning up these soils, we're really not leaving much for their potential afterwards or setting ourselves up for more work. So that kind of brings back to what FarmLab is doing and investigating. So as Diego mentioned, mycorrhizal networks in conjunction with plants work powerfully in a symbiosis of phytoremediation. So we're looking at studying the native fungi on our site, the native mycorrhizal fungi, and harvesting those, amplifying them, and introducing them to some of the plants that we've investigated that are excellent phytoremediators. And in this process, we can amplify the amount of lead we can pull out of the soil, which is essentially has a carbon footprint of 0, if not negative or positive. I can't remember which way you phrase it, but we would actually be sequestering carbon in this process.
Wow. Yeah, that's a lot.
James
has taught me, yeah, I know, it's okay. Are you kidding? James has taught me so much about fungi, and that they can do so much more than just help clean our polluted soil and water. Fungi also have a very important role, just like he mentioned, in potentially helping us address some other of our more pressing environmental issues, like climate change and desertification. So in that light, James, how do mycorrhizae and plants contribute to carbon cycling and the building of soil?
Yeah, certainly. And again, this is the thing that kind of goes under the radar, and again ties back to the theme of interdependence, because Diego and I, going back, always fight like plants, fungi, but ultimately we talk about we always talk about all the great things plants are doing to help save the world, and all the things that fungi are doing to help save the world. But ultimately, together they form one of the most powerful mechanisms for carbon sequestration. And so aside from the carbon cycling and sequestration that our oceans perform, the largest source of land-based carbon cycling, 70%, occurs between the roots of mycorrhizal fungi and plants. And in this process, basically what's happening is that the plants are photosynthesizing and taking, again, this carbon from the atmosphere, turning it into sugar, delivering that to the mycorrhizal networks, and the mycorrhizal networks are storing that within our topsoil, and transforming that and turning it into glomalin, turning it into all sorts of things, but ultimately holding it between the roots of plants and the mycorrhizal network. 70% of all land-based carbon cycling and sequestration occurs in this zone, which is why it's so important to protect our soils and to see our soils and acknowledge the living systems and soil life web, because ultimately it could be one of our greatest tools in the process of mitigating climate change and mitigating mass extinction.
Great. So we've talked a lot about the big picture things and a lot of the whys regarding FarmLab's work, but I also think that we have to go into some of the nitty gritty in regards to the how. I know it's a lot. Yeah, I know it's a lot, but can you summarize Farmlab's research into remediating lead with plants, bacteria, and fungi for our much-loved guests, and what kinds of decisions or actions you are drawing from this data?
Most definitely. Yeah, so let me actually pull up my screen here. I'll share. Uh-oh, oops, maybe I'm having a technical issue. Okay, that's actually okay for right now, but I can explain it anyway. So what we are looking at ultimately, like I said, is supporting the mycorrhizal networks in our plant communities so that we can really sort of juice up the ability of heavy metals that are being stored inside of our native plants. And then ultimately the question is, what do you do with these plants once they've kind of become compromised in the sense that they're now laced or sort of full contain heavy metals? What do we do with them? Some of the practices are to create a sacrificial pit, or the other practices are to incinerate. So the two very intense words like sacrifice or incinerate. And generally people kind of go for a sacrificial where you kind of bury it with a tree and the tree absorbs it and the tree holds onto it, hopefully for several years. That's kind of considered like one of the better practices, whereas incineration is, pretty carbon intense. So what we're looking at actually doing is taking these plant roots and chucking them in a biodigester. And a biodigester is kind of like a constructed stomach in a way that it's full of what we call anaerobic bacteria that live inside of an oxygen-free environment. And essentially letting them digest these plant materials that are laced with lead and whatnot into a kind of slurry or sludge. So again, this kind of sort of stomach action going on here. And actually the anaerobic bacteria have their own ways of dealing and mitigating with lead in the process as well. So we're getting some work done there. But ultimately, what we would be doing at the end is taking this sludgy digested plant material or even soil that's been removed in cases where we need to act more quickly, and passing it through this device we've been researching called a fungal biosorption reactor, which is a big, heavy kind of word. And I always laugh when I say it. It sounds pretty funny, but essentially what the process would be is that the skin of mycelium, and mycelium are sort of the vegetative state of mushrooms for people who don't know that, has this natural ability to kind of act like a Velcro for heavy metals, and that it doesn't get sucked up into their bodies like if we were to ingest lead. It's more like if our own skin was just really attractive to lead and it just kind of always got stuck on the outside of it. And that's what fungi have this natural ability to do. Part of that is that it's their ability to capture metals and resources to distribute and harvest for other organisms. So we would be using these kind of mycofilters to pass this sludge through. And as it's passing through, it's cleansing the sludge of these metals. And ultimately what we get at the end is this kind of composted, digested plant material that we can then use for building soil, composting, anything like that, free of the worry of, what if there's lead anymore, because we've sort of filtered that out with the fungi. And the awesome thing is at the end, we can wash off either through just simple things as baking soda, and in some cases, some very organic acids made by fungi, wash off these heavy metals and collect them and just have heavy metals. And, at that point, pass them off to the proper authority to deal with them. So we're talking about a complete removal of lead from this system. So that's very long-winded and technical, but that's essentially what we're looking into, is a way in which we can completely remove metals from our environment and cleanse our soils without having
Yeah. So I think that this is all super important, and part of what makes it even more imperative is that a lot of what we're trying to do is open source this knowledge, and we have the resources and capacity to really experiment with novel ideas and radical mycology to really solve some of the world's biggest issues. And we're also incorporating lots of different types of data, and we're super interdisciplinary in our approach. And one of the most exciting things that have come up as of late is something called cow eDNA data.
Yeah, I'm back on now.
Yeah.
Sorry about that. I don't know why I just randomly got dropped out of the call. Somebody didn't like me. I was out of the soil.
Oh, I'm glad you're back on. But
Okay.
as I was saying, James, I think it's important for us to go into some of this newer data that we're receiving. So what is cow eDNA data? Because I've worked on it like before in my undergraduate research, and even I have a hard time explaining it. So can you do us all a favor of going about it, and what's its use to the studio?
Sure. Yeah, so I'm not the most technically versed, but I'm going to kind of give a layman's understanding, I suppose. But cow eDNA is a process of understanding biodiversity by analyzing the DNA in soil, water, or different substrates. So for example, cow eDNA took samples from our site and several sites in the LA River and extracted the DNA out of it, and then processed that and matched it to the organisms that were libraried. So essentially what we're getting is, if not a complete map, but a very, very comprehensive map of all the different types of organisms, not only plants, animals, and fungi, but also bacteria, protozoa, and all sorts of other organisms that are living within our soil, have been there in the past. So it's really this incredible systems-wide view of the story of biodiversity and kind of the sign-in book of who's been there.
Cool. And what does that mean for our remediation? Like, how does that help us to connect these past living systems to what we're trying to do?
Sure. So what, in terms of remediation, what this kind of does is allow us to screen our soils for either known organisms that have been studied to, have some kind of ability to digest petrochemicals or mycorrhizal networks that are particularly adept at absorbing and working with plants to pull out lead out of the soil. So it gives us this, it kind of gives us a name and also a library of the different organisms we have in our soil that have the potential. And again, this is not only just fungi, this also pertains to bacteria as well, but gives us a big list of organisms that might have bioremediative capabilities, which we always had an intuition of, but now we have a confirmation of. And it allows us to stick with our practice and continue to work with the native organisms to our site that are primed to be dealing with these pollutants.
Cool. So James, this has been a very fabulous conversation about fungi and the role in regenerating living systems, but I think it would be unjust for us to forget ourselves in this picture, especially in these trying times. Can we easily grow mushrooms for food and medicine at home to help stabilize our food supply chains? How does the average individual go about supporting themselves and their neighbors via mushroom cultivation?
Yeah, that's a really great question because fungi are actually incredibly fast-growing organisms. We're talking about, when anybody who's gone through the process of growing mushrooms has sort of witnessed this magical appearing and disappearing. And this kind of speaks also to why they're so mysterious, is that they sort of emerge out of nowhere one day, and then within a few days after, they're dead and decomposed. And so kind of saying to that is that we can actually use, if we're trying to, everybody's thinking about our food supply chain and the security of that right now with COVID-19. We can actually use some very basic materials that are accessible to everybody to quickly and efficiently grow oyster mushrooms. So essentially all this really involves is, all you need is like a 5-gallon bucket or pot, cardboard, coffee grounds, paper, wood chips, and kind of like a sort of, you can either get it yourself from a grocery market, like the butt of a mushroom, which is kind of like the stem sort of part at the end. And essentially you can sort of lasagna those things together. And that's really all they need for food in order to be able to grow into edible and nutritious oyster mushrooms. So I just kind of want to show like a little example of like, this is essentially all we're talking about here. What I have in my hand is what we call cardboard spawn. And it's essentially just corrugated cardboard soaked in water. And as you can see, I have this mycelial network growing on it. And it's like, diffused through the entire thing. And you kind of just make these like little packet burritos of cardboard and sort of layer them together. And that's all you really need to grow mushrooms. I know I saw an article, it's an unfortunate situation, but I learned that in the crisis in Syria, a lot of people are actually growing oyster mushrooms right now and have turned to the cultivation of mushrooms because of the insecurity of their food systems over there during the war. And this kind of speaks to the ability of like, we really don't need a lot of resources and we don't depend on these sort of supply chains. We can kind of just make what we, take what we can from our environments and grow mushrooms. And so they provide this unique ability. And you don't need land, you don't need outdoor space. You really just need like a closet or a dark space and a bucket.
You hear that, everyone? Let's get growing mushrooms. So James, but I have one question. Are there any specific mushrooms that we should be growing and considering as a source of medicine?
Most definitely. So yeah, fungi, just to give a brief summary, are also play an important role with medicine. Oh yeah, let me just, because I'm having technical difficulties, let me just point this out. This here is a diagram of the research that FarmLab is doing for the remediation of lead. So I was kind of giving a long-winded explanation with nothing to look at. So if you look at the top left corner, you're seeing the plant roots and the mycorrhizal networks working together to pull out lead and channel it up into the root system and into the tissue of the plant. And then if you move to the right, we're taking this dead plant matter that's laced with lead and inserting it into a biodigester with anaerobic bacteria, turning it into this digested sludge. And then finally, we'll be feeding this digested sludge into the diagram at the bottom, which is the fungal biosorption reactor, where this digested sludge mixed with citric acid, which is actually created from a type of mold. Most of the world's citric acid comes from that. And these two things mixed together sort of make the lead more available, and then passes through this final chamber with a mycelial network where we can separate the lead from the biodigested sludge. And like it says at the end, clean organic compost for fertilizer, and we can just collect the lead for proper disposal. So thanks, John, for having my back during my technical difficulties. But yeah, back to the question about fungi and medicine. Oftentimes we forget the important role that they play. Fungi in many ways revolutionized medicine. Penicillin comes from a mold. And oftentimes many of our antibiotics come from fungi, and many of our anti-cancer sort of immune therapy, different compounds are coming from several species of fungi. So in a broad sense, fungi perform this role of immune support in that they help your system not only produce more immune cells, but make your immune cells more efficient. And we're talking about cytokines and stuff like that is a thing with particularly COVID-19 is a sort of antiviral. And again, this is kind of like giving the advice of like, these things help you boost your immune system so that you have you're kind of prepared to deal with any of the stresses and infections. It's not so much a intervention of like, you know, somebody's severely sick or anything like that. They're immunologically important to a sort of preventative medicine, but in a very clinical way that's been studied, and that they help us reproduce all of our immune cells and help our immune cells work more efficiently.
Cool. Yeah, no, James is like our resident, like in addition to being a radical mycologist, he also is our supplier for reishi tinctures. And I just like snort that stuff like left and right. And I found that it's really helped me, like, you know, it really did help me during the like peak of the flu season. And so I think it's really important that we're,
Yes.
Yeah, there it is.
So yeah, I'll just show, this is a Reishi Antler. So it's not the typical way that reishi is grown, but you know, I like to kind of show mushrooms and fungi that don't look like your typical ones, like the button mushrooms that you get. And this one is certainly like a wild, almost alien looking like fungus, but it's considered like perhaps like the crown jewel of Chinese medicine. And really is a powerful healer. I myself am a type 1 diabetic and use these kinds of things for mitigating my own disease. And another one that we have here for show and tell is lion's mane, which is a fungus that's been studied for its abilities in supporting immune systems, but also their ability to regenerate the myelin sheath in your brain, so supporting brain health over time. Yeah.
Thank you for showing me all those mushrooms. Thank you so much for all the time that you've given us, James, into the insight as to the power of fungi and what it means to be a radical mycologist. I've had a great time, but unfortunately, I think we're running out of time. So I think it would be really the best use of our remaining time to go into the second portion of our salon, which is Q&A. So please, Bees, can you take it away from me? Thank you.
Sure. Thank you so much, both of you, and thank you to everyone on the team. I'm going to start with Tilly Hinton, who asked a couple of interesting questions throughout. So I'm going to unmute you now, Tilly.
Hey, this has been a fantastic salon. Thank you. My first question was to ask whether the rapid resurgence that we've seen in air quality, water quality, creatures coming back into urban habitats, is there any similar pattern in the fungi world? Would it be realistic to expect that, James, and have you noticed any resurgences or changes in what fungus has been doing during lockdown and the pandemic?
Yeah, that's an interesting question. Well, I'll start kind of with fungi are to us in many ways invisible, not only because of our own cultural biases and our lack of education. So we don't look for them or even understand where they might be when we visually can't see them. So they're kind of always there and always working, just like bacteria are as well, except they're a little bit more visible than bacteria. But to kind of, I saw this really great meme online the other day that had this embroidery of mushrooms, and it said in the middle, it said, "You can't kill me in any way that's significant." And that's kind of the theme of fungi, is that absolutely, like with the loss of environment and the loss of habitat and the stress of everything, certainly fungi have less places to exist, but ultimately, if you kind of look at them and look at bacteria and even plants, like they're gonna survive much longer on this planet than we are. So without a doubt, they're probably definitely taking over things that are being untended right now, you know, because they're kind of sneaky tricksters like that.
So James, I'm gonna just jump in on that. Tilly, thank you for your question. Where James and I and Diego and the rest of Farm Lab have a project space on what we call the moon. During this pandemic period, I have actually observed a lot of change in the mushroom life. And I think that might partly be to do with a lot of rain that happened at the beginning of our quarantine. But certainly the living systems that we're observing in 6 different observational circles, all of them have mushrooms that are very present right now. And what I've been doing every few days when I go to visit is try and observe what the relationship between the changing temperature is, how they dry and die, or at least disappear from the surface, and what follows them, and whether there can be any anything that I can observe about that. And I would say that in the observational circles that have oak mulch, the presence of the mycelium lasts longer. Mm-hmm. And the degree that you can see the interdependence between the mushroom and the plant life that follows is more quickly visible. So It's just my observation. James, you may want to jump in on that.
No, yeah, I'd like to actually feed on that, kind of going back on, probably with people manicuring parks less and leaving debris and wood chips and fallen limbs and stuff like that. These are all food for fungi, and also, sort of the beginning process of creation of soil. So the fact that many of these things are no longer being swept away and manicured definitely means that fungi and other organisms are now free to feast as they please. And like Lauren said, like anywhere you see wood mulch, you're going to see lots of fungi. So the more we leave things untended, like kind of we are now, the more they begin to take over.
Awesome. Lois Arkin asked a couple of questions as well. I'm going to unmute you now, Lois.
Uh-oh. I don't know if anybody else is having a hard time hearing.
Yeah, Lois, we're having some trouble with your audio.
Here, maybe
I'm gonna read the question.
Yeah, that would be good.
So there's a couple of things that Lois posted that you've covered. So I'm just gonna read all of it and you can pick and choose. So James, can you talk more about fungi rendering harmless cadmium along with lead, other heavy metals? Sure. And is metabolic doing speak more to the research that we're doing on this, which I think you did touch on. And then LA needs a policy and regulation on bio/phyto/myco remediation on site. Is this something you or Metabolic Studio would be interested in working on? We're dealing with a similar situation with our brownfield here in the Los Angeles Eco Village.
Cool. Yeah, I think I saw a couple questions asking if heavy metals, like does this just pertain to lead? Does this relate to other heavy metals? And the thing is, it doesn't just pertain to lead. Fungi have a great ability at biosorbing and dealing with several other types of heavy metals, but it's not necessarily universal. So for example, there are some types of species of mushrooms or fungi that are particularly adept at biosorbing just lead, whereas you might want to look to a different species that's much more adept at biosorbing cadmium or arsenic. It kind of varies. And the same story kind of goes for plants, I think, as well. Some of them deal with and absorb different heavy metals in different ways. So there kind of does need to be research done, but also, creating resources and tools that sort of share and make this information like a little more accessible and intelligible, applicable ultimately.
Awesome. I'm going to find Carolina Goodman. I'm going to unmute you now for the next question.
Hi, everyone. I typed my question a while ago, so I don't remember. Can you read it? Sorry.
No problem. Who else is using all of these processes for cleaning soil? So who else is out here in your network?
Well, I definitely know a few people that I've made connections through at the UC Riverside Department of Environmental Toxicology. they do really great work. Some of the people from there have an organization called Co-Renewal, and they're also looking into understanding some of these processes. And yeah, I mean, I kind of also just, for me on my like mushroom level, I know that the studio's done work, and maybe Lauren, you can talk about maybe some other people that we know.
Well, also, I think Carolina's question when it was coming in was in regards and specifically to the work that you're doing for the studio in remediating the lead that's going to be coming out of the ground. And if that's correct,
Okay.
Carolina, one of the things that James and I and the rest of the Bending the River Back into the City team have discovered is that there isn't many explorations going on about how lead can be remediated from construction site material and moved in a lead-free way to other landfills. So what we're hoping to do with our work building an infrastructure sculptural object is to explore cleaning all of the soil that we pull out of what used to be a Standard Oil site and remediating it using phyto and mycelial cleansing, and trying to open source how that can happen because of the ubiquitous nature of lead-based problems in construction soil. James, do you want to say anything more about that, about what the state of the art kind of?
Sure. Yeah. So I would say probably most of this, like probably 2 parts of the system we talked about today have been done by other people. So definitely people work with mycorrhizal fungi in companionship with phytoremediating plants and try to amplify and support that relationship. And also people are using a biodigester to break that material up. But ultimately, usually where the step goes is that you take that biodigested sludge and you kind of bury it around a tree so that the tree sequesters the lead for many years. Like I kind of said, it's the biosorption reactor is kind of the thing we've been exploring because it really hasn't been done before, at least not to scale or not really outside of academia. So we're trying to take a leap here. And sort of in the spirit of radical mycology, which is making all of these concepts and ideas and practices of mycology more accessible, more so that we can disseminate them and open source them, take this concept and practice and turn it into some way that can be community organized and even applied by individuals or organizations or communities.
And a second part of Carolina's question was, can mycelia cleansing help with the polluted aquifers that lie beneath Los Angeles County? And it is our strong belief that the interdependence of the mycelial network and its relationship with bacterial and plant remediation all working together is an absolutely certain way to cleanse the pollutants out of polluted aquifers. The main question is the function of time. How long will that take? And can we stop making the pollution problem worse by extracting more water from the water table, making a higher concentrate of pollutants in the water that is there? James, do you want to weigh in on that at all?
Yeah, I just want to say I concur, and essentially, like, there is no doubt that these organisms together can deal with this, at least chemically and enzymatically. But the question is, how do we do it? What's the carbon footprint of it? What's the repeatability and accessibility of it? We don't want to just bust open an aquifer with machinery and start tinkering with it with all these doing all these things to it. We kind of want to think of a design and a process that, as we refer to in remediation as in situ, insight without excavation, that can work over time with the least amount of impact on our aquifers and our living systems.
Great. There are a lot of really good targeted questions, but I'm going to skip to Jasmine Sarp, who I think summarizes some of these. And I hope that we can have James stick around and answer some of the other specific ones. So Jasmine, I'm going to unmute you now.
Hello, everyone.
Hey, Jasmine.
My question is, do you have recommended best practices for citizen mycologists who are interested in applying mycoremediation in public or natural spaces that are difficult to monitor on a day-to-day basis? And is there a way to filter contaminants in these spaces while avoiding the development of contaminated fruiting bodies that animals or humans might unknowingly eat?
Yeah, that's a tough question, because oftentimes that's the trick. I would say it's a big question. It kind of needs to be divided into sort of two categories, because you really have to separate chemicals like petroleum from heavy metals. So I would say that if you're looking at dealing with heavy metals in soil systems, if you want to work in environments that have been disturbed, or even environments, that are by disturbed, I mean more kind of like in an urban habitat. I would say working with activating and supporting the mycorrhizal fungi is a great way to cajole that process and give it life and give it what it needs, so that you don't need to be there to monitor it. And ultimately, like, the fungi will deal with these heavy metals in a way that makes them less toxic to plants, and also therefore makes it less toxic to us and organisms that eat the plants. In waterways, that's again kind of tricky. You can kind of build mycelial filters that you insert into waterways, but you also sort of need to understand like a time frame for how long you can leave it there before you got to go get it back. But to that, I would say, again, mycorrhizal fungi work with, are incredibly important to aquatic systems and wetland systems as well. So again, working with that relationship, being conscious of that relationship is important. And lastly, about chemicals. Chemicals, you don't really need to worry about like the idea of like a mushroom that pops up that's full of heavy metals or something like that that you might eat, or a plant or an animal might eat and suffer from lead poisoning or something like that. And that is because the process of dealing with chemicals for fungi and bacteria is actually a destructive process. The fungi are actually digesting and eating and metabolizing the petrochemicals, because they're very similar to the kind of structures they eat within wood, and therefore their enzymes have the ability to deal with that. So ultimately, at the end of a process where fungi and bacteria go through a 7-course meal of the different types of petrochemicals, really what you're left with in the end is just carbon and water. And that just goes straight back into the nutrient cycle and feeds back into the life web.
Thank you.
Mm-hmm.
Awesome. And we have time for one more question, and I'm going to call on Rachel Neubauer. Rachel, I'm going to unmute you now.
What was the question? Because I asked a couple.
What is the one action a person can do or take to support mycorrhizal fungi and mycelial networks in their own yard or communal property?
Thank you, Bees.
Yeah, that's a really good, and I know you said one thing, but I'm kind of gonna give like 2 or 3 because it's complicated. But one of the things you can do is not adding fertilizers to your lawns or fertilizers to your gardens, because mycorrhizal fungi only form relationships with plants when they accept them. And the reason plants accept them is because they need the networks to provide them with nitrogen and phosphorus. So oftentimes in hydroponic systems, when you're kind of like dumping in a bunch of phosphorus and nitrogen and stuff like that, the plants are saturated with it and don't need to form that relationship. And the mycorrhizal fungi just kind of die off because they are, like I said, interdependent on forming this symbiosis. So that's one way. And the other way is kind of like working with no-till farming, or trying to disturb the soil as least as you can once you've built it to a certain point where these organisms are well established. It's literally like kind of like ripping their skin off when you do that. It just like exposes them, and you know, it stresses them out. So that's definitely one way you can work with mycorrhizal fungi is no fertilizers, limit the amount you till on your land.
Thank you so much, James. Thank you everyone for your incredible questions. If you guys can imagine me listening to Diego and James on a daily basis at Farm Lab, it's pretty incredible. Thank you again, you guys. So I'm Millie, I'm also from Farm Lab, and today I'm gonna give you guys a couple of shout outs. We wanna give a shout out to Alma Farms, who is in Compton, California. They will be distributing 200 free grocery kits, which includes fresh and prepared foods on May 10th. So I would look them up if you're in need of some fresh food or vegetables. To our other partners, Northeast Trees. They're an incredible nonprofit organization here in Northeast LA. They transform gray landscapes of Los Angeles into beautiful landscapes, and they've planted over 100,000 trees in the past 20 years here in Los Angeles. So thank you so much, Northeast Trees, for being our partners. Also, another shout out to Urban Homestead, who are located in Pasadena. If any of you guys are in need of a CSA box or looking for fresh produce, I would definitely tap into their website. Thank you again, and back to Lauren.
Thank you, everybody. Thank you for attending the second Interdependence Salon. Again, we are interested in using this Zoom platform to emulate the mycelial network and realize that we're all in this together, and we're all, in a way, exploring a brand new way of consciously being in the world by sharing our resources, sharing what we grow, share how we think about how big is here. So when we talk about our garden, realize that there's systems underneath your garden which don't rely on property lines. You, by supporting the mycelial network, you also defeat the whole idea of private property, because it's the network that you're supporting. And also to think about what it could mean in Northeast Los Angeles in particular, which is cut up by every highway in Los Angeles except for the 405, which is got the geological time floodplain sealed over by massive amounts of concrete, which we've discovered may have actually protected it from exposure to all kinds of pollutants during the last 100 years. So if you think about protecting the mycelial layer underneath the concrete that still thrives and still existed, then in a way you are building the soil body of the post-pandemic world. And it should give you a feeling like it gives me of hope to have the Farm Lab team support our commons, to support the building blocks of life itself, our soil, our seed, our water, and our community process. So thank you for your brilliant questions and for being present. Before we close, we're going to have our studio team member, Lou Pesce, who lives alongside the LA River, who gives the most extraordinary tours of the floodplain and all things connected to what will be the distribution network of 106 acre feet of cleansed LA River water, hence his t-shirt, Water Right number 21342, which is the studio's independent water right. Why that's important is that we're looking to activate this citizens' utility through these conversations to make sure that we become the stewards of the life systems that we need moving forward. So over to you, Lou.
Thank you so much, Lauren. As we have been talking about remediation and making a difference along the LA River, we thought we'd close today with a poem by someone else who has made a significant difference in the LA River. That is Mcadams. He is a poet. He was a poet. and the founder of FOLAR, Friends of the Los Angeles River, and he was a staunch advocate for the Los Angeles River. So this is a poem from his River Cycle, poem number 20 from book 1. And although it was written in 1998, it is even more poignant now after his recent passing. It's called Old Man of the River. For years I thought I was going to find the old man of the river, a guru who knew everything there was to know about it. He was going to draw me a map. Then I'd know what to do with the information that was written in my soul. I never found him until I caught my reflection in a puddle of rainwater. It was me all along. Bushy brows, balding skull, worried eyes, in awe of 8 egrets as they lift into the evening. Thank you.
Thank you so much, Lou. Just a moment of silence to remember Louis Mcadams and what a great poetic mind brings to the possibilities of the future still emerging. Rest in peace, dear Louis. To close, I just wanted to say we at the studio will stay on to say goodbye to all of you. So as we end the session, if you want, John will unmute everybody, and please feel free to hang out, talk amongst yourselves. We'll hold the space as long as there are people here. And
Yes, I think so.
please feel free to join us next. Please feel free to join us next Thursday for Interdependence Salon number 3 with Dr. Vandana Shiva, who will be speaking with us from India. I will be in dialogue with her, and we are very happy to be collaborating with our colleagues in Brooklyn, the Brooklyn Rail, on their format, the New Social Environment, which they have been hosting daily at lunchtime, New York time, 1 to 2 p.m. Pacific Coast time, 10 to 11 daily. Today they had Noam Chomsky speaking, and they're doing amazing, amazing work. And in many ways, our Interdependence Salon is modeled on what they are doing. So they've kindly offered to host our conversation next week with Dr. Shiva, because her time difference is 12 and a half hours away. So it's way easier for her to join us at 10 to 11 next Thursday, 10 to 11 Pacific Standard Time. So thank you, Brooklyn Rail, and please join us. You can sign up on Eventbrite for that talk. And yeah, now we can unmute you and chat on your way out.
Yes.
Bye. Thank you very much, James.
Thank you, James.
Bye.
Thank you, Diego. Thank you, Lauren.
James.
James, everyone.
Love you, James. You rock.
Thank you, guys.
Thank you.
Brilliant, James. Brilliant.
Thank you, Lauren.